Okay. Hi, Miss Sloan. And this time I promise we are going to do chapter 7, cellular respiration. So let me get it started here. Okay. And here is, can you do these items? So kind of read through that a little bit. Let me move me. Over here. So just read through it. What ones do you think you need a little help on? What ones do you have a good grip on? And then I just popped in the additional objective right here to remind you of it. And if for some reason you don't have your book and you want to take a picture of mine on that page, you totally can. All right, are we good to go? Can I move forward? All right. So first of all, overview, okay? When we talk about cellular respiration, we always want to remember, and we'll review the song as well, who does it? All cells, okay? All cells do cellular respiration. Now, they may do it aerobically or they may do it anaerobically, but all cells do cellular respiration. Because in essence, what that's going to do is take the energy that's locked up into glucose, which is a potential form of energy, right? And convert it into a form that's spindable in our own body cells. And what spins well in our body cells? ATP. And the first law of thermodynamics says that energy cannot be what? Created or destroyed. It can just change forms. So we're changing that form of glucose into a form of ATP. Now, we also know the second law of thermodynamics says every time you change forms, you lose some energy as heat. So you don't have as much free energy, right? You're losing some of that energy. Of the two, aerobic or anaerobic, which one maximizes the transfer of energy? Aerobic, right? The use of oxygen, okay, and doing all the steps of aerobic respiration maximizes the amount of ATP we can get, which is the sole point of cellular respiration, is to generate ATP. Anaerobic respiration is useful for those organisms that are unicellular and only need a little bit of ATP, or for like us, we do it as backup. When we're struggling and we don't have quite enough oxygen, we'll do anaerobic respiration as backup. Okay, what's universal about these? Everybody does the first step. And what's the first step called? Glycolysis, right? And we know that there are 10 different steps, 10 different enzymes, and we'll talk about that. But here we make ATP. And that's the next part I want to review is the three different ways. Now that you've learned photosynthesis as well, you know the three different ways to make ATP, right? Okay. Substrate level phosphorylation is that direct handoff, like what we do in glycolysis, where I have the phosphate and now I'm giving the phosphate to you, having an enzyme catalyze that process. That's substrate level phosphorylation, direct handoff. This way, oxidative phosphorylation, this is your na-na-na-na-na-na-na, and oxygen is at the end, right? Catching the electrons at the bottom of the electron transport chain in cellular respiration, right? And it's removing the electrons so the chain can keep moving, because as long as the chain keeps moving, we're doing reductase, help me out here, reductase, cytochrome oxidase. We're putting hydrogen ions out in the intermembrane space. And if they're out there in the intermembrane space, you have a difference in concentration. What else? A difference in, not temperature, difference in concentration, pH, and charge. Okay? Difference in concentration, pH, and charge. And that difference, that gradient, then hydrogen wants to come back in, and it can come back through ATP synthase complex, and you can make some ATP. Okay, substrate level is a handoff. Oxidative phosphorylation, you're using electron transport chain, and oxygen's at the end, keeping the electrons having the ability to move through the chain so you can make ATP. Photophosphorylation is what we learned about in photosynthesis, right? And we talked about how light is at the beginning, right, to excite the electrons. So you can do a na-na-na-na-na, right, in photosystem 2 and then again in photosystem 1. So you can make some ATP, yes, and some reduced NADP. But photophosphorylation is talking about photosystem 2, that first one. Okay, do you feel comfortable with the 3 different ways of synthesizing ATP? Is all ATP made the same way? No. Okay, good. All right. So then the other thing we need to remember with cellular respiration is we need to get comfortable with the parts of the mitochondria. Okay. So if you wanted to do this and you wanted to draw on here again, you could, right? And I don't know if I can draw and do loom. I will try. Will it let me? No. Okay. But if I was going to write on here, I'll just write it this way. Where is the intermembrane space? Out here, right? This is the intermembrane space. Is it going to let me write? No. Okay. So this is your intermembrane space right here. The matrix is this. Oh, there it goes. The matrix is the absolute center, right? And then the cristae is this folded part right here. Okay. So where are hydrogen ions concentrated? A difference in charge. What else? pH, concentration, right? Okay. What else did we say? Okay. Where are they concentrated? Out here in the intermembrane space. Okay, remember when we said reductase, cytochrome oxidase, we are pushing the hydrogen ions out into this area. Okay, where does the electron transport chain? Right here on the cristae. That's where the electron transport chain is. Okay, bunches of them. Okay, where does the Krebs cycle and transition prep take place? Right here in the matrix. Okay, questions or concerns? I can see you guys drawing and you're doing a great job. Any questions on the anatomy? Okay, what is the first step of cellular respiration called? Glycolysis. Where does it occur? It does occur in all cells, and where? In the cytoplasm. Okay, so you don't have to do anything with the mitochondria in order to do glycolysis. It is 10 steps. It is 10 different enzymes, right? But ultimately what you're doing is you're taking your glucose and you're breaking it in half. And when you break it in half, you will end up with 2 3-carbon units. Glucose is a 6-carbon unit. You'll end up with 2 3-carbon units. What is the name of these 2 3-carbon units? Yeah, pyruvic acid, okay, or pyruvate. That's another name for it. And along the way in those steps, you will reduce some NAD, and you need to reduce some NAD to do the next part, which is what? Make some ATP. If you don't reduce the NAD, you can't make any ATP. So that means if you're reducing it, you have to start out with some NAD in the oxidized state, right? And we called NAD, we gave the example of being a mule, right? So it's like an empty mule with a full mule, okay? You're reducing it with those, transferring those electrons, okay? Now, glycolysis in and of itself will just get you 2 ATP. Is that enough to keep us alive? No. So we've got to do more with it. So what we'll do now is we'll go ahead and take these 2 pyruvic acids, the end product, and we'll go ahead and do, what's the next step in aerobic respiration? Transition prep, right? And we're going to move into our mighty mitochondria, and we're going to take our 2 pyruvic acids, which have 3 carbons. We're going to pull a carbon off each one, and when it comes off as what? What does it come off as? Carbon dioxide. So here we can see the 3 carbons here of pyruvate, right? And then we're going to pull off CO2. There it is right there. Since we have 2 pyruvates, we'll have 2 CO2s. And when we do that, we'll pull out some of the energy, some of the electrons here, and we'll use that to reduce some NAD. Okay? This CoA right here jumps on right here. Okay, it jumps on in that spot. And why is that important? Because what's the next step after transition prep? What's the next step after that? Krebs cycle. This CoA gets us in the door to be able to do the Krebs cycle. No CoA, if we don't have that coenzyme, we can't go into the Krebs cycle. So that's why it's important. Does transition prep make us any ATP? No. Do you see any ATP getting made here? No. Does it make us anything good? Two reduced what? NAD. That's good. We can cash those out later for some ATP, right? And it makes us 2-acetyl-CoAs, which we can feed into the Krebs cycle. Do we like the Krebs cycle? Yes, because it makes us a whole bunch of NAD and FAD, right? Mostly NAD. All right, so following transition prep, we go into our Krebs cycle. We will do the Krebs cycle 2 times because we have 2-acetyl-CoA. And preloaded, and remember, when I have C2, C4, C6, all of that, I'm just having you follow the carbons. I'm not having you don't need to know how many hydrogens or oxygens or anything else. But preloaded in our Krebs cycle is a C4 molecule. Acetyl-CoA is a 2-carbon molecule. So we know when you add a 2 to a 4, you're going to get what? 6. So we have this 6-carbon molecule. We peel off 1 carbon as CO2. 6 minus 1 is what? 5. In the peel off, just like in transition prep, when we pull off that CO2, it allows us to reduce an NAD molecule. Okay? And then we're going to do that. And when we do that, what did we call that in our song so we would remember it? Do the bit. Okay? And we're going to do the bit again. We're going to pull off the CO2. We're going to reduce an NAD, and now we're down to 4. Is there still energy in this 4-carbon molecule? Yes, because we're able to do a substrate level phosphorylation of ATP. We're able to reduce a new player here, FAD, and we can still reduce another NAD. Now it's low energy. And so we're going to hook it up with a second acetyl-CoA and do this whole thing again. The Krebs cycle doesn't make us any ATP. Yes. How many ATP does it make? 1 per turn. And how many times are we going to do this? Twice. So when we're done with the Krebs cycle with our 2-acetyl-CoAs, we will have 2 ATP. Did we make any ATP in glycolysis? Yes. I know you're smarter than you're appearing right now. You know the answers to this, right? Reduce some NAD and make some ATP too, right? You make 2 ATP in glycolysis. Do you make any ATP in transition prep? No, you just make a couple NAD, right? Do you make any ATP in the Krebs cycle? Yes. How many? 2. So all of that ATP making we've done so far is all substrate level phosphorylation. It is literally passing a phosphate group from one molecule, the ADP, to make it ATP. That's all it is. And we don't have very much. Where you really get the ATP from aerobic respiration is our next step. What is our next step? Electron transport chain, okay? So when you do the electron transport chain, you're going to take your NADs, who get, remember when we said gather all the reduced NADs and FADs and take them to the top of the ETC, right? You are going to take your NADs and they get to go to the top floor here. They drop off their electrons. Those NADs that were reduced, once they drop off their electrons, they become what? Oxidized. Be with me, children. I'm worried right now. They become oxidized. Why is that good? Well, they dropped off their electrons, right? So we can do the electron transport chain. But why do we want oxidized NAD? Yes. So I can reuse them in glycolysis, transition prep, and the Krebs cycle. There's a finite amount of NAD molecules. So if you want to keep using them, they got to be able to unload their electrons. Okay, and they're going to unload their electrons when they get oxidized at the top of the electron transport chain. Okay, as the electrons move through the electron transport chain, they go from high energy to what? Low energy. But along the way, they give some of their energies to the molecules that are holding them, specifically reductase, cytochrome oxidase, who can use that energy to take hydrogen ions from the matrix and put it into the intermembrane space, right? We want that to happen because the more hydrogen ions we have in the intermembrane space, because the only way they're going to get out there is by reductase, and cytochrome oxidase. And if we can keep those hydrogen ions out there in concentration, they'll want to come back what? In. And they come back in through ATP synthase complex, and now I can generate ATP. That's where I make a lot of my ATP. For every strong NAD I bring, I will get 3 ATP. Now tell me, rank these in order of energy. ATP, NAD, and glucose. Rank them, talk to the person next to you. Rank them energy. ATP, NAD, and glucose. So rank them. All right, come back to me. ATP is just one ATP, right? One point. And NAD can get me how many ATP? 3. That's more energy, right? Because I can get 3 ATP for every NAD. Glucose gets me 36 to 38 ATP. Okay, because if I do cellular respiration, the process of cellular respiration will convert that glucose molecule into 36 or 38 ATP, not accounting for the second law of thermodynamics. Okay, but could get me 36 to 38 ATP. Are you following that? Okay, so I want as many NADs as I can get because those will get me 3 ATP each. FAD, not as powerful. FAD comes in a little lower, so it doesn't contribute as much to the hydrogen ion gradient, so it only gets me what? 2 ATP. Okay, you all right with that? Okay. Now, that math I did right here. Okay, so you can see glucose to pyruvate, substrate level phosphorylation, 2 ATP. And now this is transition from 2-pyruvate to 2-acetyl-CoA. That's transition. Okay, does that get me any ATP? No. I'm going to do the Krebs cycle twice, or the citric acid, same thing. I will get 2 ATP, substrate level phosphorylation. Where I really rake in the ATP are gathering all the reduced NADs and FADs and take them to the top of the ETC, dropping off their electrons, and then I can cash them out for ATP. And I can only do that because the final electron acceptor at the bottom of the chain is oxygen, which will then form water. No oxygen, then I don't have any removal of electrons out of the chain. I won't make my hydrogen ion gradient. So look right back here. Okay. So at the bottom of my chain, if I want to keep these electrons going so I can keep the hydrogen ions generated, then I need some oxygen to remove those electrons. I have an indoor and I have a what? Outdoor. I got to keep the chain moving. Okay, more hydrogens out, the more ATP that I make. Okay, questions or concerns about that? All right. So that was the hardest part. All right, then here, this is just telling you proteins. You can digest proteins. You can pop them in at pyruvate. You can pop them in at acetyl-CoA or citric acid cycle. So it's not just glucose and carbohydrates that do cellular respiration. Proteins can help you with that, and fats can help you as well. All right. And then what happens, though, if I don't have any oxygen? If I don't have any oxygen, I can do the glycolysis part, but without oxygen, I'm going to run into trouble because I have a finite amount of NAD, and the NAD gets reduced through the process of glycolysis. The sole reason you do fermentation, it's not going to get you any more ATP. All it does is it acts the same way that the electron transport chain acts, in that it oxidizes NAD. NAD drops off its electrons back into pyruvate, thus actually reducing the pyruvate either into alcohol or lactic acid, just so you can reduce them again in glycolysis. You have to empty the mule. The mule has to unload, right? So you can use it again. And the reason is you always have to reduce some NAD before you make some ATP. So that's why you do fermentation. Okay, got that? All right. So then this right here was just another summary, okay, kind of an overview of what happens, okay, showing you the two. Remember, you and I, we do lactic acid, whereas plants and yeast, they will make alcohol as a byproduct of their fermentation. And then this was another review right here as well. Okay, I have some practice questions in here if you want to do them, but do you want to do the song one time to make sure you have it? Okay, so let me try to do this. Maybe, baby, I love you. Okay. Sorry, delay of game. Hopefully it'll work. Okay, am I in the picture? Okay, here we go. What's the whole thing? Do you want me to remove this so you could, does it matter? Okay, because I'm far enough away from you. What's the whole thing called? Cellular respiration. What's the first step called? Glycolysis. Who does it? All cells. Where do they do it? In the cytoplasm. What do you start out with? Glucose. How many carbons? 6. What are you going to do? Break it in half. How many steps? 10. How many enzymes? 10. What are these called? 2 pyruvic acids. And you reduce some NAD and make some ATP. 2. Oh, we have plenty of oxygen. What should we do? Aerobic respiration. What's the next step called? Transition prep. Here we go. And you reduce some NAD and 2 acetyl-CoA, put 1 away. What's the next step called? Krebs cycle. What's another name for it? How many times are we going to do it? Why? Because we have 2 acetyl-CoAs. Ready? Oh, what does do the bit mean? Reducing an NAD and pulling off a CO2. Okay, good. 2 plus 4 is 6. Do the bit, get 5. Do the bit, get 4. Make an ATP, reduce an FAD, reduce an NAD, and 2 plus 4 is 6. Do the bit, get 5. Do the bit, get 4. Make an ATP, reduce an FAD, reduce an NAD, and stop. Gather all your reduced NADs and FADs and take them to the top of the ETC. Reductase, cytochrome oxidase, and you make some ATP. A lot of it, a lot of it, a lot of it, or a lot of it, a lot of it. Who catches electrons at the bottom? Forming water. Give me your glucose. Break it in half. How many steps? How many enzymes? What are these called? And you. Oh no, we don't have any oxygen. What are we going to do? Fermentation. How many choices do we have? 2. You and I make lactic acid. Makes our muscles sore. Plants and yeast make alcohol. Either way, you oxidize your NAD. Okay, feel good about that? Okay, do you want the practice questions? Okay. All right, here we go. Hopefully this, there, is it going? There you go. I'm going to control it from right here. Okay, what's your answer here? Is all ATP made in the presence of oxygen? No, the answer is false. Good. Okay, riddle me this. What's your answer? True. Good. Those are easy. Okay, so what do? They transfer electrons, the energy from the glucose to the what? ETC, the electron transport chain. Good. Okay, riddle me this. Pardon? Well, yeah. Yeah, so what's your answer here? True, right? For cellular respiration. Good. All right, and here? Oh, we do not agree on this one. Does the glycolysis require ATP? Yes, 2 to get the party started. If you don't have any ATP originally, you're not going to be able to do it, right? 2 to get the party started. Then when we do it, we get how many back? 4. So we net out 2. Okay, so the answer is true. Okay, riddle me this. Does glycolysis require oxygen? No, it does not. What part of cellular respiration actually requires the oxygen? Who catches at the bottom? Oxygen forming water. What did we just do? Electron transport chain. But if you don't have oxygen, you can't do the electron transport chain, which means you also can't do the what cycle? Which also means you can't do transition prep. Okay, good. All right, next. Okay, what's your answer on this one? True. Yeah. Okay, because you're either making lactic acid, that's what you and I make, or you're making alcohol and CO2. Okay, are you saying something about that fermentation oxidizes the NAD so it can be reused again during glycolysis? Okay. It oxidizes the NAD so you can reuse it again during glycolysis. Okay, and riddle me this. Maybe, baby. There we go. We are not in agreement with this. Check with the person next to you. Does glycolysis generate any carbon dioxide? No. Fermentation does. Transition prep does. Krebs cycle, that's it. Right? Okay. All right. And you got this. Any questions? Anything you want to ask me? Yes.
How does it move from anaerobic respiration if it doesn't do the water?
No, you've reached the end. You're either making lactic acid or you're making alcohol. For us, the lactic acid can be converted back to pyruvate when we're not in oxygen debt, and then it can go through the oxygen way for us. But some organism, that's the full stop. That's the end. Oh, I have to teach you that. Yeah. All right, study, make good choices. You're welcome. Yeah, it's right over there.